Hello everyone, I'm Priyanka and today we will discuss the estimation test for lactate dehydrogenase. Name of the test colorimetric method. Talking about the principle, here the lactate dehydrogenase will catalyze the reaction. The reaction is as follows: lactate plus NAD will produce pyruvate and NADH2. Here, the lactate dehydrogenase activity can be determined by estimating the concentration of pyruvate in the reaction. Now, here the 2,4-dinitrophenylhydrazine react with pyruvate in an alkaline medium, giving brown color 2,4-dinitrophenylhydrazone. Color intensity is proportional to the concentration of the pyruvate, which give measure of lactate dehydrogenase enzyme. Here requirements are test tubes, surgical pipettes, thermostatic water bath, photoelectric colorimeter. Here we will discuss the reagent composition. First we have substrate solution. For substrate solution, first we require glycine buffer 120 ml, zero point 0.1 normality of sodium hydroxide solution will be 20 ml, 35% weight by volume sodium lactate solution will be 10 ml. Method of preparation: will take 120 ml of glycine buffer, 20 ml of zero point 0.1 normality of sodium hydroxide solution and 10 ml of 35% weight by volume of sodium lactate solution in 250 ml volumetric flask. Then we will dilute it to 250 ml, adjust pH to 10 with zero point 0.1 normality of NaOH solution. You can store this reagent in refrigerator. Next we will discuss the composition of glycine buffer, sodium lactate solution and sodium hydroxide solution. For the glycine buffer, we require glycine 3.753 grams, sodium chloride 2.922 grams, distilled water up to 500 ml. Dissolve almost 3.753 gram of glycine and 2.922 grams of sodium chloride in about 350 ml of distilled water and dilute to 500 ml with distilled water. Storage: store the solution in refrigerator. Next we have 35% weight by volume sodium lactate solution. For this, lactic acid will be around 52 ml, sodium hydroxide 21.5 grams, distilled water up to 200 ml. React to 25 ml of lactic acid with 21.5 gram of sodium hydroxide, dissolve in 120 ml of distilled water by boiling for about 30 minutes. Avoid bumping using broken glass pieces. Cool and make the volume to 200 ml with distilled water. We can store the solution in the refrigerator. Next we have 0.1 normality of sodium hydroxide solution. For this, we required sodium hydroxide 0.4 gram, distilled water up to 100 gram. For the method of preparation, dissolve around 0.4 gram of sodium hydroxide in about 75 ml of distilled water and dilute to 100 ml with distilled water. Standardize the solution and adjust normality if necessary. Stability: this reagent is stable at room temperature for several months. Next we have NAD solution. For this, we require nicotinamide adenine dinucleotide 10 grams, distilled water 2 ml. For method of preparation, dissolve 10 mg of NAD in 2 ml of distilled water. Storage: store this solution in refrigerator. Store solid NAD also in refrigerator. Next we have sodium pyruvate concentration solution. For that, first we require sodium pyruvate 220 mg, glycine buffer up to 100 ml. Dissolve 220 mg of sodium pyruvate in 75 ml of glycine buffer and dilute 200 ml with glycine buffer. Next we have sodium pyruvate working standard solution. For that we required sodium pyruvate concentration solution around 5 ml and glycine buffer up to 100 ml. Method of preparation: dissolve about 5 ml of sodium pyruvate concentration solution in 100 ml of glycine buffer. Stability: this reagent is stable at 2 to 8 degrees Celsius. Next we have the 2,4-dinitrophenyl hydrogen. For this, firstly we require 2,4-dinitrophenyl hydrogen, almost 200 mg, and concentrated HCl 85 ml, distilled water up to 1 liter. Method of preparation: here we will dilute almost about 85 ml of concentrated HCl to about 750 ml of distilled water, then dissolve 200 ml of 2,4-dinitrophenyl phenyl hydrogen in the diluted acid solution. Dilute the solution prepared in step B, that is dissolve 200 ml of 2,4-dinitrophenyl in the acid solution. Now we will make up the volume by using make up the volume to 1 liter by using a distilled water. Talking about the stability, this reagent is stable at 2 to 8 degrees Celsius. Next we have 0.4 normality of sodium hydroxide solution. For that, firstly we required sodium hydroxide 1.6 gram. Next we require distilled water of 100 ml. We will dissolve 1.6 grams of sodium hydroxide in 75 ml of distilled water and dilute to 100 ml with distilled water. Standardize and adjust normality if necessary. Talking about the stability, this reagent is stable at room temperature for several months. Here in this test field sample will be a serum sample. One thing is needed to be noted down, that is red cells contain almost 150 times more activity of lactate dehydrogenase enzyme than in serum. Hence, the hemolyzed serum should not be used for the lactate dehydrogenase determination. So, the wavelength at which we will measure the absorbance will be around 505 to 510 mm. We will use green filters here. So, next, talking about the procedure. Label two tubes as test and blank and pipette into them as follows. Next we have the concentration tables. Okay, so here we have two tubes, test and blank. First of all, we will add a substrate solution, 1 ml of substrate solution in both test and blank. Then we will add NAD solution 0.2 ml in both the tubes. Then keep them in water bath for about 5 minutes at 37 degrees Celsius. Then we will add serum in the test solution 0.1 ml, and after that 15 minutes of incubation at 37 degrees Celsius. Then we will add 2,4-dinitrophenyl hydrogen in both the tube, test and blank, that will be around 1.0 ml in each tube. Then we will add serum sample in blank, around 0.02 ml. After that 15 minutes of incubation at room temperature. Then we will add 0.4 normality of NaOH in both the tube, that will be around 10 ml. Mix thoroughly and read OD value of test and blank against water. Here we will subtract the absorbance of serum blank from the OD of the test. Determine OD of the standard as follow. For the absorbance of standard, Take two tubes, label them as standard and blank, pipette into them as follows. Firstly, we will add the substrate 1 ml, then water 0.2 ml in standard and 0.3 ml in blank. Then we will add pyruvate standard in the standard tube. Then we will add 2,4-dinitrophenyl solution in standard and blank both the tube. Concentration will be around 1 ml. Then keep in 37 degree for 15 minutes. Then we will add 0.4 normality of NaOH in both the standard and blank tube. That will be concentration will be around 10 ml. Okay. Next, determine the absorbance of standard against your black. Talking about the calculation, activity of lactate dehydrogenase enzymes in serum will be equal to OD of test minus the OD of blank divided by the OD of standard into 333 international units per liter. If the enzyme activity is more than 1000 international unit per liter, dilute the specimen and multiply the determined value with dilution factor. Talking about the normal value in adults, this value be around 70 to 240 international unit per liter, and in children it be around 150 to 590 unit per liter. So that's all we have in the determination of lactate dehydrogenase. Thank you and have a nice day.